Let's wait a few seconds until the presentation is up in the screen. While we wait for the presentation to appear on the screen, I would like to say that it is indeed an honor to be here in this conference today and speak about my own relationship with DMT. I come from a community in the Amazon who's been using chacruna. We've been long-term users of ayahuasca, and that's why those sacred plants really caught my attention from the very beginning. That is the topic of my brief presentation today. It is not a topic that's very much dealt with in the Congress. However, it is very important because the ayahuasca tea that we take is made with these plants. So taking care of these plants is indeed very, very important so that we can drink quality tea. Now, this work, that what I'm going to show you, is a part of my PhD thesis that I am currently working on at the Sao Paulo University in Brazil. I have the support of the National Institute of Research of Amazon. I am a civil servant there. I'm a biologist myself, and also have the support of UDV that has let us enter into certain areas to collect samples, plant samples. We know that P. viridis, which is our chacruna, is original of the Amazon rainforest, and it is a rainforest, so high indexes of rainfall, and there we include other biomes such as continental land, savanna ecosystem also, and other ecosystems that we could mention like swamps and marshes. We are going to deal with two environments, continental land and the savanna forest in the north in Brazil. That is when it was first grown in the north of the country. Then we started to grow chacruna in other regions in Brazil, nearly all the regions in Brazil, with the different climate traits and characteristics that, of course, varied from one place to another. When the plant is not in its native state, in its native context, we may start to detect morpho-anatomical variability in the plant, and this supports the appearance of different ecotypes that are new. These new ecotypes usually are linked with physiological differences that are established within the same species, the same plant species, and those physiological differences may lead to variability in the chemical compounds of the species. And that's when we start to wonder what happens with P. viridis in different regions in the country. In 2018, our collaborators conducted a study about chacrunas grown in different areas of the country, and they understood that there are chacrunas in different regions that present different amounts of DMT produced. And why is that? Why does this happen? We tend to link this to the environment working in the Amazon, and in the Amazon what really interferes with the plant characteristics is the high intensity of light and water pulse, rainfall pulse, because we have two seasons in Amazon. Another one which first one which is very, very rainy, another one that's less rainy, but rainy indeed. And we focused on chacruna. We grow in western Brazilian Amazonia. You have to hear the two regions marked in colors. The one in yellow, we could say, is the savanna area. That is what the landscape looks like, and the land proper inland in the area of the Amazon. The savanna is characteristic by lowest annual rainfall, and inland is characterized by higher annual rainfall, higher than in the savanna. Indeed, we collect the samples at the end of every season, because when we collect a sample, a leaf at the end of the season, the leaf itself has been produced during that season. So under the environmental climate conditions of that season, which will leave a trace, an imprint in the anatomical and morphological characteristics of the leaf that we are going to study. Since we will be talking about morphological characteristics and anatomical characteristics, let's see what this means, what we mean by this. To prepare ayahuasca, we use the leaves because it is well known already that the leaves contain the highest amount of DMT compared to the whole of the plant. If we perform a cut, here is a section view to release DMT or to find it, a plant that's well cared will show these characteristics. If it's well taken care of, we see the duct there that will be formed, domate, and we see this dark line that is the central innervation, and that corresponds to this part here in the leaf. This is another section, the limbic cut, that is the limbus here, this part of the leaf. So we have this region, the cuticle. It's the first line of defense, anti-stress for the plant. This layer here is the epidermis, as if it were the skin of the leaf, and underneath we have the same structure. And in between the two of them, we have the region where all the photosynthetic machinery of the plant is located, and that is where the photochemical metabolism takes place, hence DMT production. The changes in this region, lead to alterations or biochemical variability in the plant, as I suggested earlier on. The leaf pattern is determined by the environment and by the leaf biota. Here is a leaf. So in the end, we will find this region that looks like a smaller leaf, but it's called an apical stipule that protects the beginning of the formation of the plant, which takes place right here at the center. That's the beginning, the very beginning, the birth, made up of this little dents, made up of a compound, gel-like compound. Here are the names of the parts, and there's some little bugs that we will talk about later on, some bites that actually cause the formation of those domatia that we call the guide for chacruna. But in this place there's a small opening. You see them here, and these are DMT cells deposited there like a repository. And here within, inside this place, that's where these little mites live. That's where they live, and those mites are very important because the chacruna really shelters those mites in this part of the organism. Let me skip some slides. About the leaf, when we take a look at the leaf, we see that in the chacruna leaf there's some wax which is covered by fungi. You see those filaments, those strands there are fungi strands, which are very necessary for DMT production. DMT is, for us, indeed, the compound that creates the activity, the effect of ayahuasca, as if one were drunk. And who attacks this part? Those components here, those fungi that inhabit, let's say, part of this region. What is it these components do, these fungi do? The mites are omnivorous, so that is why there's a mutual reaction. The chacruna shelters them, and then they can do their job. When we see this sort of wax in the chacruna, we see that there's different deposits, inland, savanna, but on the inland there's not that much sun. They don't receive that much sunlight. However, in the savanna there's less water, more sunlight. And given the land, the soil characteristics, it's different. You see those little ducts here, those little notches cause chacruna to cause reflectance, so reduce the intensity of solar radiation that reaches the leaf. So let me skip some slides. Here we see the first line of defense of the leaf under stress, or water stress, high insulation, high levels of sunlight. The chacruna shrinks the leaf as in an adaptive mechanism. So it's a strategy to reduce biotic stress, because when this happens, when this adjustment happens, it reduces the activity of the fungi at that very precise moment. So this is also a strategy to increase the shelf life of the leaf. When we see all these characteristics, all these morphological adjustments, we can see the movements inside the leaf. Anatomical changes have taken place to make these adjustments, and those anatomical changes will lead to alterations that then may lead to biochemical alterations and changes in general. When the leaf suffers those movements, the ducts need to be generated, and these ducts take the whole length of the leaf. So it's not about rolling up the leaf, it's not just that, but if it's like if it were a hose, because when the plant feels the presence of water, it can deploy and open up much more quickly without having to proceed with the habitual moves. Another line of defense is the reduction of the area taken by the leaf, so that leads to stomatal density reduction, and that's when gas exchange occurs for photosynthesis. In order for the leaf to shrink, to become smaller, the density of the leaf also is reduced. And that leads to increased thickness of the cell. So density is increased and thickness is increased. The intracellular spaces they reduce, and then we generate water economy in the leaf. And at the same time, there is low mesophyll conductance in the area where the photosynthetic machinery is located. Reduced photosynthesis then, and this means reduced biochemical production. Another characteristic of the chacruna is the creation of these structures inside that you see to the left, the image to the left-hand side. These are druses. These are calcium oxalate crystals that help the leaf reflect the high reflection, but the production costs leaf, and that cost is reflected eventually in the production. Here you see the image that clearly shows what I am referring to. This leaf was produced on the inland in a moment where water needs were quite low. So you see that in yellow or orange color, those are the alkaloids in the leaf, and that was produced in the same period, low rainfall, but in the savanna. And here to the right, you see that DMT is a defense line together with alkaloids, and they're located in specific areas, especially in the region, intracellular region, preventing fungi from entering this region here. Here's stomata open to the right-hand side, while they're closed in the image to the left, so nearly no DMT. That's why we can conclude that water pulses, prolonged water pulses in the Amazon, and their capability of water retention induce morphoanatomical alterations that are directly linked to the photosynthetic metabolism, and thus this can cause a variation in the DMT production. Thank you very much for your attention.